Hello and welcome back to Biology Explained with me, Mr. Hill. In today's episode, we are going to be looking at the cell membrane structure, as well as permeability, diffusion, osmosis, and active transport. This is Unit 1, Key Area 2 of the National 5 Biology course here in Scotland. But of course, if you're joining us from anywhere around the world, not doing the SQA National 5, then this could still be very useful for your revision for biology, say at GCSE level across the world. If you like this video, please do hit like and subscribe to the channel so you don't miss a future episode, and do make sure you tell your friends who are also doing biology who might need that G up and might need that improvement in confidence ready for their biology examinations. Okay, so without further ado, we're going to make a start by looking at the cell membrane structure. Okay, so welcome to the first part of this episode where we're going to be looking at the structure of the cell membrane. Now, when you're drawing out your cell membranes on your cell diagrams, you usually just use a single line because at the cell level, that's all you see is just one single line. But if we zoom in to the molecular level, you will notice that the cell membrane is in fact made up of two layers. And these layers are made up of a molecule known as a phospholipid. Now, a phospholipid is a fat molecule. That's where the word lipid comes from. That contains a phosphate group, hence why we call it a phospholipid. And the phospholipid arranges itself into two layers. So you've got a double layer of phospholipids, and we call that a phospholipid bilayer. So on this diagram here, as you can see, I've got the green heads and blue tails. Each of these individual units is a phospholipid, and so we can label this up as our phospholipid bilayer. Phospholipid bilayer. Okay, and at National 5 level, you just need to be able to name it. But you'll notice that the tails are always pointing inwards and the heads are always pointing outwards. And that's because the tails are hydrophobic. That means they don't like water. Okay, and so they kind of hide themselves away from the outside world. And the heads are hydrophilic, philic meaning to like, and so the heads are always pointing outwards towards the water. And that secures that double layer membrane and makes it really tight and secure and acts as a barrier that allows things in and out. Okay, so that's the phospholipid bilayer. You'll also notice on our diagram here that we have two different other molecule types here. Both of them are proteins, but they do different things. Okay, so the orange ones here we have proteins that are found on the surface of the cell membrane, and these proteins have a variety of different roles. They're often used in cell signaling. Okay, and receptors. So they are responsible for picking up, say, hormones that are floating around, and then when they pick them up, they'll trigger something to happen inside the cell. So our membrane then has these proteins on the surface, but they also have proteins that cross the membrane, okay? And we call these transmembrane protein. Transmembrane protein. Trans meaning to go across. Okay, so it goes across the membrane, and by doing so, it creates this channel. Okay, sometimes we call them channel proteins, and this channel is there to allow certain molecules to come in and out of the cell. If you remember, the cell membrane's function from our last episode is to control what goes in and out of the cell, and it does that through the use of transmembrane proteins. Okay, so these will open and close, allow different molecules to go in or out of the cell accordingly. Okay, now it's important to notice that the cell membrane is fluid. Okay, so it's kind of moving around like a sea. Okay, and the proteins kind of float around them. Okay, now we call this the fluid mosaic model, and the fluid mosaic model is what you see in front of you. Okay, so the fluid mosaic model. Okay, this is stretching a little bit for the N5, but the fluid mosaic model is how we describe how these proteins sort of move around the membrane. The membrane itself is pretty secure, okay, with the phospholipid bilayer, and the proteins are kind of floating on the sea of it. Okay, now that's important to think about when we think about what might affect how permeable the membrane might be, and we're going to look at that in our next section. So welcome back. In our first section, we looked at the fluid mosaic model and the structure of the cell membrane. Now we're going to have a look at this idea of permeability. Now, a membrane is said to be described as being selectively permeable, okay, or partially permeable. And selectively permeable means that it allows certain things through, but not everything. Okay. And the degree in which a membrane can allow something to pass through it is known as permeability. Okay, so there are different environmental factors that can affect how easily molecules can pass through a membrane. And if it has a high permeability, then it means it allows a lot of things through. If it has a low permeability, it means it doesn't let many things through at all. And remember, membranes are selectively permeable, so they have a limit to what they can allow through. And that's important for the cell to be able to control what comes in and what comes out. Okay, now, one factor that we need to look at that affects the rate of permeability is temperature. Okay, so temperature is one of these very common factors that affect many biological reactions and processes because biology is very sensitive to temperature. And permeability is no different. Now, you can see on the screen, I've got two boxes here, describe and explain. We are going to answer this question, how the temperature affects permeability. Okay, and we're going to be looking at describing it first of all, and then explaining it. Now, this is an important exam technique. Okay, describe means that you're saying what happens, and explain is saying why. And it's very important that if you're asked a question to describe something, that you are telling me what happens rather than giving a reason. And if you're asked to explain, that you need to be saying why, not just saying what. Okay, and this is something where students often, miss a couple of marks here or there by not going into detail with the explain section. So let's have a think at what happens then as you increase temperature and to the rate of permeability or to the degree of permeability. So we're going to describe first of all. So to describe the effects of temperature on permeability, all you can say is that the permeability will increase. Okay, so permeability increases as temperature increases. Okay, that's me simply describing what happens. So if you had a cell and you increase the temperature of the solution that it's found in, then it's going to start to allow things in and out of the cell a bit more quickly, okay, because it's becoming more permeable. You've described it. Now, the reason for this is all to do with kinetic theory. Okay, so thinking back to lower down the school when you've looked at how particles change their behavior as they gain more energy as they heat up, it's the same principle here in biology. So as you warm up a membrane, that fluid mosaic model that we looked at is going to start moving around a little bit quickly, a little bit more quickly. They're going to vibrate more, the molecules are going to vibrate more, and by doing so, it becomes a little bit more gappy, okay, a little bit more permeable. Well, yeah, naturally, it's going to be a little bit more leaky, and so molecules are going to be able to pass through. The molecules themselves, of course, are also moving more quickly, and so they are more likely to find that transport protein, that transmembrane protein, to move into or out of the cell. So let's write down our 2 points then as to why an increase in temperature increases permeability. So the first step is that there's an increase in kinetic energy. Okay, and kinetic energy is related to how the particles vibrate. So increasing kinetic energy increases fluidity of membrane. Therefore, molecules are more likely to pass through. Second point is that the molecules themselves are moving around more, so they're more likely to pass through a transmembrane protein. So molecules more likely to pass through a transmembrane protein. Okay, and both of those reasons are why then an increase in temperature increases the permeability of a cell membrane. Okay, so once we've done that, we're now going to start looking at how molecules can pass through a membrane through 3 different processes: diffusion, osmosis, and active transport. So we have now looked at the structure of the cell membrane and discussed this idea of permeability, that membranes are selectively permeable, and the degree of permeability is dependent upon environmental factors such as temperature. Now we're going to start looking at how molecules can actually move through a membrane. And there are 3 processes: diffusion, osmosis, and active transport. And there are similarities and differences between all 3 of these. So we're going to start with the most basic. We're going to start with diffusion. And diffusion is a type of passive process. That means it does not require energy to occur. Okay, this occurs just naturally, and there's no kind of active process involved in the movement of molecules by diffusion. So let's make a note of that first of all. Our first key point then is that diffusion is a passive process. Okay, no energy is required for this to occur. Now, you will see that across this membrane, I've got two concentrations of molecule. The purple is the molecule here. And you can see on the left-hand side, okay, we have more molecules than we do on the right-hand side. In diffusion, the molecules are simply going to move from the area of high concentration to an area of low concentration. So molecules will have a net movement, so they're going to move around kind of randomly, but the direction that they're going to be moving in as a whole is going to be from here, where it's high concentration, to here, where it's a low concentration. Okay, so that's our second key point to say. Okay, so it is the movement, and I'm going to just write shorthand here, movement of molecules from a high concentration to a low concentration. High concentration to low concentration. Okay. We say that it's down a concentration gradient. Okay, so you might see this written down a lot. It's down a concentration gradient. Okay, imagine a high concentration is at the top of the hill and a low concentration is at the bottom of the hill. And the greater the difference, the steeper the hill, the faster the diffusion is going to occur. Okay, so diffusion then, very simple. It's passive. It's any molecule. It doesn't actually need to occur through a membrane. So if you spray some deodorant in the corner of the room, the scent will diffuse across the whole room. It doesn't need a membrane. But in biology, it often does, of course, occur across a membrane. And it's a movement of molecules from a high concentration to an area that is a low concentration. Okay, so examples of molecules that might diffuse are small molecules only. Okay, anything large needs a transmembrane protein. Okay, or might have some other more complicated processes, but diffusion of small molecules will occur just straight through the membrane, and that will include things like oxygen, okay, into, say, for example, into a red blood cell at the lungs, okay, so into red blood cells at the lungs could be an example of oxygen diffusing into a cell, or molecule diffusing into a cell. Okay, so that's diffusion. We're now going to have a look at our next passive process, which is going to take a little bit longer to describe and explain, and that is osmosis. So see you on the other side. Okay, so now that we've looked at diffusion as one of the simple passive processes of movement across a membrane, we're now going to look at our second passive process, which is a little bit more complicated, and that is the process of osmosis. Now, osmosis is a passive process just like diffusion. Okay, so it does not require any energy to occur. However, osmosis relates to the movement of only one type of molecule, and that is water. So it's the movement of water only. Okay? And you can see on our diagram here, I've got two different concentrations of a solution. The blue circles here are representing the water. Okay? And I've got some purple one here that might represent some salt of some sort. Okay, so something that might be dissolved in the water. Okay, we're just going to label it salt. Osmosis is concerned with the movement of water across this membrane, not of the salt. And water will always move from an area of high water concentration to an area of low water concentration, down a concentration gradient. So it's the same description and definition as diffusion. Only this time we are concerned with water. So if you look at the diagram here, we can see that we've got quite a dilute solution here, meaning there's quite a high water concentration, and quite a concentrated solution here, which has a lower water concentration. Now that's where sometimes it can get a little bit confusing when we are describing and explaining osmosis. So let me just explain that one more time. A solution that has a high water concentration is quite dilute. So if we were to visualize what this solution might look like in a beaker, there's not going to be many, not many salts at all. And so it will look a little bit like this. Okay, see here, quite a dilute solution, quite watery, not very concentrated. Here, on the other hand, we have a lot more salt, and so if we were to visualize what this beaker might look like, okay, we can see it's a lot more concentrated. Okay, imagine instead of salt, this might have been Ribena. Okay, other squash is of course available, and you would notice that if you were to make your squash concentrated, it will look a nice dark purple here, compared to a more dilute one, which is quite watery there. Okay, so we can call a high water concentration is equal to a dilute solution, whereas a low water concentration is equal to a concentrated solution, because there's a lot more salt there relative to a dilute solution. Okay, so this is a concentrated solution. Now, in the National Fives, you will see it written as a high water concentration and a low water concentration. But in other literature and other places you might get in some of your revision, you might see dilute and concentrated solutions. So it's important that you can that you are aware of the different ways that we describe these solutions. So in osmosis, then, water is going to move from a high water concentration to a low water concentration down a concentration gradient. So water is going to move in this direction, the net movement of water. Okay, so remember, molecules move at random. Okay, so they go sort of everywhere. But the net movement of all the water will be moving in that direction until the concentrations are the same. Okay, so let's make a note of that. Okay, it's the movement, and again, I'm just going to write shorthand here. Okay, it's the movement of water from a high concentration of water, okay, we call that a dilute solution, to a low water concentration, okay, down a concentration gradient. Okay, one last thing though to mention is that it does require a membrane for this to occur. So water is going to move from the high water concentration to a low water concentration through the membrane. And if you remember from our last part, we described the membrane as partially permeable or selectively permeable. So you need to mention that as well when you're describing osmosis. This occurs through a selectively permeable membrane. Okay, so to recap then, osmosis then, it is a passive process, does not require energy, and it's the movement of water molecules only from a high water concentration to a low water concentration, down that concentration gradient through a selectively permeable membrane. Okay, so take a breather, and then when we come back, we're going to be looking at how different cells respond when put into solutions that are higher or lower water concentrated. Be right back. So, welcome back. Now we're going to be looking at the effect of osmosis on cells according to what solutions they're placed in. Now, this is a very common area of questioning in exams to see if you can apply your knowledge of osmosis to different situations. And you'll have to be aware that different cell types, animal and plant cells, will behave differently when placed in solutions that are either more dilute, so a higher water concentration, or more concentrated solutions, lower water concentration. So we're going to sketch this out on this diagram here. Feel free to pause the video and get yourself ready, and pause as we go through to make notes on how the different cells behave when placed in different solutions. We're going to start by looking at animal cells. So, for example, I'm just going to draw myself a red blood cell, okay, in the middle here, and a red blood cell in the middle here. Now, all cells have a concentration of salts in them, okay, 1, 2, 3, 4, 1, 2, 3, 4, and they have water in them as well, of course. Okay, so 1, 2, 3, 4, 1, 2, 3, 4. Now, in a solution of higher water concentration, if you put this animal cell here, so these are my water molecules. Okay, imagine this was pure water in this case, okay? Or it could be at a concentration that is more dilute than inside the cell. If you put the cell into this pure water or this lower, this more dilute solution, water is going to move by osmosis from outside the cell to inside the cell. Okay, so water will move inside the cell from outside. And that's going to cause the cell to swell, okay, as it fills with water. Now, animal cells, they don't have a cell wall. So if they swell too much, they're eventually just going to pop and burst, okay? And so that cell will die, and you will have no cell. So let's make a wee note of that. When an animal cell is put into a higher water concentration solution, okay, water enters via osmosis. Again, be really explicit if you're ever asked to describe this. Say it's by osmosis. That's going to get you a mark. Okay, and then the cell will swell and burst. Okay. Now, on the flip side, if you have an animal cell and you put it in a more concentrated solution, okay, so I'm going to add my water here again. But this time, imagine this was a really salty solution. So I'm going to add lots of our salts here. So it has a lower water concentration than inside the cell. Water is going to move by osmosis from inside the cell to outside. Okay, so the opposite direction here. Water will move out of the cell and into the solution. Okay, so that's our first point that we need to make. So water leaves the cell by osmosis. Don't forget to say that. Okay, by osmosis or by osmosis. And what happens is the cell is then going to shrivel, and it will be sort of shrivelly, right? It's not going to burst, okay, because it's the opposite from the other process. Simply, the cell will shrivel, okay? Now, it has a term. This is known as being crinated, but in National 5, you're all right. You don't need to know that term. So all you can just say is that the cell shrivels. Okay? The cell shrivels. And so that's what happens to the animal cell. Now that we've seen the effect of different solutions on animal cells, now let's have a look at what happens to plant cells. So we're going to complete the boxes again, but this time with the plant cells. And so in the middle, I'm going to draw ourselves some nice plant cells. Now remember, plant cells have a cell wall and a cell membrane. So I'm going to draw a nice angular cell here, because remember, plant cells are typically more angular. And then we are going to draw our cell membranes there as well. Okay, and just like in our other scenario, our plant cell will have a concentration. Okay, so we're going to add some water. We'll add some salts. Okay, these could be anything that's dissolved within the cytoplasm or the vacuole in this case. And if we would imagine that we put our plant cell into a solution that has a higher water concentration, i.e. it's more dilute. This could be pure water in this example. So lots of water molecules. Water, just like before, is going to move into the cell by osmosis down that water concentration gradient. Only this time, the cell is not going to burst, and that's because plants have cell walls. Instead, the cell is going to swell up, that vacuole is going to swell up, and it'll be nice and firm, and we call that turgid. The cell becomes turgid. Okay, we looked at that last episode, but it's important that we note it in this context too. So if we were to write up what happens, okay, we have water enters via osmosis, just as before. Okay, only this time the cell swells and becomes turgid. Turgid. Now, when a cell isn't turgid, we call it flaccid, and when the water sort of swells into the vacuole, we call it turgid. And that's when the cell is nice and firm, the plant is standing nice and tall, and that's what we want to see our plants to be like. Now, if you put the plant cell into a solution that is more concentrated, i.e. a lower water concentration, so a very salty solution in this example. Okay, so lots of salt here, and let me add some water molecules as well. Then, as before with the animal cell, water is going to leave the cell via osmosis. And the membrane will start to shrivel. Now, because the plant cell has a cell wall, the cell wall is still going to be intact. The membrane, however, is going to peel off that cell wall, okay, and sort of shrivel within the cell wall itself, okay. And so if I sketch just outside the box here, what it looks like, okay, you're going to have your cell wall here, but the membrane is going to shrivel within it. That has a term. We say that the cell has become plasmolyzed, okay? So the cell membrane has peeled off the cell wall and shrunk within itself. It's become plasmolyzed. Okay, so if we were to describe the process, water, okay, leaves by osmosis. Okay, so let me just draw those arrows in for good measure. So water is leaving by osmosis. Okay. And the cell becomes plasmolyzed. Plasmolyzed. Okay. So there we have it then. When you put animal cells and plant cells into solutions that have a higher water concentration or a lower water concentration, they'll behave differently. And we've just described those different processes. So now over to the last part of this topic, and that is to look at the process of active transport. Now, before we looked at diffusion and osmosis, they were both passive, meaning that no energy is required and the molecules are moving from a high concentration to a low concentration. But sometimes in our bodies and in plants, we need molecules to go in the opposite direction, from where they are a low concentration to a high concentration, essentially going against the concentration gradient. So if you imagine the high concentration was at the top of the hill, a low concentration at the bottom of the hill, then it's like trying to push a rock up to the top of the hill. Okay, it's going to require some energy for that to happen. And that's exactly what active transport is. So rather than being a passive process, it is in fact an active process. Okay, it requires energy. And energy comes in the shape of ATP. Now, ATP is a molecule we find all over biology that is made in processes like respiration and is used as a kind of currency by cells to spend on, and it's an energy currency, okay? So ATP is used by cells for things to happen, okay? And active transport is an example of where ATP is needed, okay? So ATP is our molecule that provides the energy for active transport to occur. Now, if we're to describe the process, we'll say it is the movement of molecules from a low concentration to a high concentration against a concentration gradient. So let's write this down. So it's the movement of molecules from low to high concentration. I'm just going to write shorthand here, okay? And it's against a concentration gradient. Okay, so if we were to look at our diagram here, we will see that it is moving in the opposite direction to our diffusion process. Okay, so here we've got a high concentration on the left and a low concentration on the right. And if we want more molecules to go left, we're going to have to use ATP, and it's going to have to be active transport. Okay, so ATP is used, and the molecules will be moving across against a concentration gradient. Now, an example of this that might happen in real life could be the absorption of different nutrients and minerals by plants from the soil into the roots. A plant might be full of nitrates already, but if it needs a little bit more, it's going to have to use active transport. So for an example, could be the absorption of, say, nitrates in soil to roots. Okay, so where there's already plenty of a molecule within a cell, but it needs more, it's trying to get as much as it can, it will actively draw it in using the process of active transport. Okay, so we've covered then our membranes, diffusion, osmosis, and active transport. Now it's time for a little question. So it's question time now for the episode, and today's question comes from paper 2 from the 2019 SQA National 5 biology paper. As you can see, this question involves a little bit of data interpretation and a little bit of calculation. This isn't that uncommon for looking at cell membranes and transport. Okay, so get used to doing some mathematical questions when it comes to this topic. It's also very good practice to be doing this every time you revise. So whilst you spend some chunk of time looking at notes, making mind maps, watching this video, that you do also spend time looking at questions so you get used to the type of command words that you will experience, as well as understand the timing and the sort of answers that may be needed for you to score your points. So there are 4 marks here available, looking at different types of ions as they move in and out of cells. Why don't you pause the video, have a go, and come back in 5 minutes' time and we'll have a look at the answers. Okay, so how did you do? Let's make a start by looking at the answers to these questions. So the first question then, 2A, is asking us to name the process by which sodium ions move into this cell. So we can see that the concentration is higher outside the cell than it is inside the cell. So if it's going to be moving into the cell, it's going to have to be going from a high concentration to a low concentration. So this must be diffusion. Okay? We know it's not osmosis because it's not water, and we know it's not active transport because it's going from a high to a low concentration. Next question. So potassium ions move by active transport. Use the information in the table to complete the diagram by drawing an arrow to show the direction in which the potassium ions move. Okay, so we know that it is moving by active transport. So this must mean it's going from a low concentration to a high concentration. And the low concentration is outside the cell, which means the molecule must be moving into the cell. So an arrow on our cell here, for the K+, moving from outside to inside, is how you're going to score your mark. Next question. Name the type of molecule found in a cell membrane which is involved in the movement of ions by active transport. So think back to that first section. We had our cell membrane, our phospholipid bilayer, and there were different molecules embedded within that membrane. Those were transmembrane proteins, okay? And it is transmembrane proteins that are responsible for movement of ions into the cell by active transport. Now, you will get a mark if you just said proteins, okay? So you don't need to say transmembrane, but it's always good practice to be as specific as you possibly can. Last question then, some basic maths. Calculate how many times greater the concentration of chloride ions is outside the cell compared to inside the cell. So we've got 116 outside the cell, and we've got 4 inside the cell. So it's quite simply 116 divided by 4. Now, you'll have a calculator with you in the exam, so you can just plug it into your calculator. But if you're doing it mentally, like I am, 100 divided by 4 is going to be 25, and then 16 divided by 4 gives me 4, so the total there is 29. It's 29 times greater outside the cell than inside the cell. So there we have it. We've covered a lot of ground today, looking at the cell structure of the membrane, as well as diffusion, osmosis, and active transport. If you have a question about any of these topics, please feel free to write them down in the comments below, and I'll do my best to get back to you. And if you have liked this video, then please check out my others and subscribe to the channel so you don't miss any future episodes. Likewise, if you know anybody else who's studying biology at National 5 here in Scotland, or indeed around the world, then do let them know. If they need a little bit of support, then they can come along and watch this video too. Okay, so it's a goodbye from me, and I'll see you for the next episode. Take care now. Bye-bye.